Hello, my name is Olivia Haddadine. I will be presenting my research on species differences in retinal A2 connectivity. I did this research through the Jones Lab of the Moran Eye Center. Animal models are commonly used in an attempt to better understand human biology and disease. Studying the similarities and differences between species can help us choose the most appropriate model for research. In the presence of daylight, cone photoreceptors absorb photons. In the absence of daylight, however, rod photoreceptors absorb photons. Despite having different types of photoreceptors, the latter part of both of these pathways is identical. This is shown in the red boxes. This is where the A2 cell comes in. The A2 cell is unique to mammals. It allows signals sent from rod photoreceptors to follow the same pathway as signals sent from cone photoreceptors when traveling to the brain. The A2 cell uses conventional synapses to communicate with off-cone bipolar cells, and it uses gap junctions to communicate with on-cone bipolar cells. These two types of output are the focus of my research. In order to research retinal connectivity, cross-sections of eye samples are taken, and then TEM, or transmission electron microscopy, images are captured. We then go through all of the sections and look for different occurrences. We mark these occurrences, which is called annotation. These 4 images show things that we might look for. Conventional synapses and gap junctions are the focus of my research, which are in the left column, but there are also different occurrences such as communication through ribbon synapses or structural components such as adherence junctions. Once cells have been annotated, they can be reconstructed using a 3D rendering program. This image shows 3 A2 cells from the rabbit. They have been annotated and reconstructed. The red, blue, and yellow dots are communications between cells. My research is focused on conventional synapses and gap junctions in A2 cells. Gap junctions are going to be yellow in this image, and conventional synapses are blue. On the left is the rabbit A2 cell, and on the right is the mouse A2 cell. In addition to breaking down output by conventional synapse and gap junction, I decided to break down each type of output by the cell they communicated with, which is called the partner cell. Here are 2 images that show the breakdown of conventional synapses by their partner cells. Partner cells for conventional synapses include amacrine cells, off-cone bipolar cells, and ganglion cells. The rabbit A2 cell is on the left, and the mouse A2 cell is on the right. These 2 images show the breakdown of gap junctions by their partner cells. Partner cells for gap junctions are on-cone bipolar cells and A2 cells. Once again, the rabbit A2 cell is on the left, and the mouse A2 cell is on the right. This figure summarizes my research. Gap junctions were the most common type of output for both rabbit and mouse A2 cells. For conventional synapses, the off-cone bipolar cell was the most common type of partner cell in both species, but the second and third most common type of partner cell differed. For gap junctions, the most common type of partner cell also differed between species. This shows that there are species differences in retinal A2 connectivity. I hope to discuss this with you further, and thank you for watching.